catalysis. This is going to contain a lot of organic review, which is why we take an extra day to try to go through this material. Maybe a couple extra days to go through this material. Okay, we've tossed this word around a lot. Enzyme. That is a protein catalyst. There's another word I want to mention that we might come back and talk about if we have time. Ribozyme. That is an RNA catalyst. Okay, for the longest time, it was thought only proteins could act as catalysts until they discovered ribozymes. Some general information about enzymes. Your reaction rates are about 10 to the 6 to 10 to the 12 times faster than an uncatalyzed reaction. Okay, and so most chemical reactions in our cells, most all of them, are catalyzed by enzymes. They're going to be several orders of magnitude faster than chemical catalysts. Okay, so there are other catalysts out there that are not biological in nature. So they're not proteins, but there are some other kind of catalysts. So sometimes it's a surface, sometimes it's an alternate mechanistic pathway that you're offering, but your chemical catalysts tend to be slower than the enzymes. The enzymes are better, they're faster. And again, that's going to vary depending on what system you're looking at. Another thing to keep in mind about enzymes is that your reaction conditions are usually mild. When you're looking at some of those organic catalysts that you talked about back in organic chemistry, I don't know. Can someone give me an example? One we just talked about was Lindlar's catalyst to get stopping at alkenes when you're trying to hydrogenate an alkyne. That's what we're doing in class right now. Do you remember what kind of conditions we're usually doing those under? High heat, high pressure. Yeah. So, and depending on what type of reaction you're trying to do, it could be ridiculous pressures. It could be hundreds of times atmosphere for your pressures, and then temperatures that are nowhere near a nice 37 degrees Celsius like you would consider most biological things. So your reaction conditions are mild compared to chemical catalysts. So that includes temperature, which could be like room temperature or body temperature. So we just put something down around 37 degrees Celsius. So it's not thousands of degrees. Pressure is just going to be pretty much atmospheric for most things. Here's another one. pH. It's going to be somewhere around neutral. So you don't have to put in these crazy acids and things in order to make things happen. No concentrated sulfuric acid needed. And so just in general, you're mild and solvent. What's your solvent going to be? Water. I hope it's water. Okay, so I don't have to dig out the methylene chloride to make this reaction happen.
If the enzymatic catalysts are usually around neutral pH, does that mean it's all happening in the blood rather than inside organelles?
No. It's closer to neutral than compared to, so like, for example, in organic where you have to have concentrated sulfuric acid and your pH is like negative something, we're within a neutral range within a few pH units. Yeah, so anything from like 2 to 3 up to 11, 12 is considered physiologically relevant.
Oh, okay.
Yeah. second great thing about enzymes is that you have high specificity. And I'm going to put usually here, because there are exceptions to this. For both the substrate, which is the molecule acted upon, and the product form. Okay. Usually that specificity is much greater than it is for any kind of chemical catalyst. And another great thing about enzymes is that these are usually reversible. And again, it's a usually, so there are lots of incidents, there we go, where you have an enzyme that's forced to be irreversible, but it's usually set up in terms of how much substrate you have going in versus product coming out and how fast the rate is, and not necessarily because the reaction itself cannot go backwards. Okay, so we're going to see examples like that too. Some brief things about nomenclature. We're not going to waste our time learning how to name things. That's the good news. The bad news is that we're going to use common names for almost everything, so you just have to know what they are. But you can usually recognize enzyme names because your names end in, you should remember this, ASE, ASE. So if it ends in an ASE, it's an enzyme. There are common and systematic names. That tell you something about what they do. And in parentheses, I'm put maybe. If it's a systematic name, it's going to tell you something. It's a common name. It may or may not tell you something. So, for example, amylase. That's one we've talked about. If you didn't know what amylase did and you saw that name, what might you infer? It does what? Do you have any idea whether it's breaking down amylase or building up amylase? Or modifying amylase? Or so what can I know? I might know that it acts upon, not amylase, but amylose, right? I might be able to infer that it acts upon amylose, that might be the substrate, and that's about all you can tell from the name. Not much of anything else. This is the same one, so this is a common name. Here's a systematic name. You're not going to learn how to do this, but it's useful to be able to interpret it a little bit. So 1, for alpha D glucano hydrolase. Okay, that is then your systematic name for the same enzyme. So it tells you more. What does it tell me? It's a hydrolase. Yeah, so I'm breaking bonds with water, right? So it tells me it's a hydrolase and I'm breaking some sort of bond. What else does it tell me? Yeah, so, there we go. So I'm breaking a 1,4 glycosidic bond. Okay. And what am I breaking? This is my substrate, which is a polymer of D-glucose. And like I said, we're not going to learn how to specifically write systematic names, but if you're presented with a systematic name, you should be able to pull a little information out from it, like what kind of enzyme it is, what it's doing, and what is my substrate. So systematic names are more useful that way, but this isn't like organic where it's super easy to learn how to name everything systematically. Yeah.
Why is it glucan?
I don't know. I'm cutting between two glucose residues, so some hydrolases, depending on what your polymer is, it might cut. So let's say it was a lactose, that your beta galactosidase. In that one, then it ends up being, it says a galactose galactoglucano or something like that, where you know that it's cutting between a galactose first and then a glucose going from non-reducing to reducing it. So there's a reason for it, but like I said, we're not going to worry about trying to do anything with that other than maybe a little interpretation. And most importantly, that you know, to interpret what that last word is. So there are 6 classes of enzymes based on the reactions they catalyzed. The order actually does matter here. So number 1 is an oxidoreductase. What kind of reactions is that going to catalyze?
Redox.
Yeah, redox. These are all redox reactions.
Why does the order matter?
I'll let you know in a minute. Yes, oxidoreductases are class number 1. So those are redox reactions. Some subclass names that fall under this category include oxidase, oxygenase, dehydrogenase, reductase, and that's probably not all of them, but that's just a few examples where they, rather than using the major class name, they might use a subclass name to describe it. Class number 2 is a transferase. What do you think it's doing?
Transfers the functional group.
Transfers, yes, transfers functional groups. And this is important between 2 different molecules. Usually. Clarify that. Sometimes you'll see transferase used with isomerases, and that's bad form. You probably shouldn't, but people do it all the time. So technically, if you're an isomerase, you're an isomerase, you're not a transferase. So should be between 2 different molecules. Third one is hydrolase. Okay, we already said that was breaking bonds with water. And to try to make this clearer, this is not additions of water across double bonds. That is something else. Usually when you have hydrolysis, you're going to break a molecule into 2 pieces. And then I have to put in parentheses my usually. So we talked about our saponifiable fats, our esters and our amides. Those are things that can be hydrolyzed, and you're breaking it into 2 different pieces. And number 4 is a lyase. Lyases are sometimes hard to identify because they're probably the most diverse in terms of the types of reactions we see. So, if you're in doubt and you can't figure out what something is, it might be a lyase. That's usually how it works. These are elimination and addition reactions. Okay, so that includes the addition of water across a double bond. But I have to put a not addition of H2, and why is that? It's a reduction. Yeah. Okay, so you just have to be careful that you aren't calling some sort of oxidoreductase a lyase by mistake. You have to make sure that you're not considering gain or loss of hydrogen or oxygen. This often involves breaking of double or single carbon-carbon bonds. And with hydrolases, you're usually breaking something else that's more labile. So again, those amides and esters tend to be acted upon by hydrolases, but if you're breaking a carbon-carbon bond, it's usually a lyase. Yep.
Looks like lyases tend to form double bonds.
Yeah, I hate the book's example because they're wrong. They're only talking about one particular subclass of lyases. Yep. So the book's definition is a little bit misleading because there are lots of different addition and elimination reactions that fall under the class of lyase. There are some subclass names that you should know to help recognize some of these. Decarboxylases. So, here's an example where this is an elimination, but it's actually a carbon double bonded to oxygen that you're forming because it's an elimination of a CO2 molecule. But in that elimination, you actually end up breaking a carbon-carbon bond in the process. So we will see examples like this. We're going to do one. If not Wednesday, then Friday. Dehydratase. And then if you go the other way, hydratase. Okay, so that refers to elimination or addition of water to form or break a carbon-carbon double bond. That's number 5. It's isomerase. What's that one going to do?
Make isomers.
Makes isomers, yep. So this is going to convert between isomers. Now those could be stereoisomers, geometric isomers, or constitutional isomers. So there's lots of different, any kind of isomer class that could include. So with that stereo, geometric, constitutional, it's just going to be interchange between two isomers. A subclass name that you should know, mutase is one that's frequently used. And then this one is the one you have to be careful of. Intramolecular transferase. So technically, people should not be calling isomerases transferases. They are incorrect to do so, but occasionally when you're reading an article, people do it on accident. And they're really referring to an intramolecular transfer. But if you swap two functional groups on the same molecule, it's really just an isomer of what you originally started with. So technically it's an isomerase and not a transferase. Okay, and last one is a ligase. This is usually the easiest one to spot. This is bond formation between two molecules to join them together. And it usually requires ATP. So it's an energy consuming reaction, and you will usually almost always see ATP being consumed during the reaction. Okay, so lots of examples today. First we're just going to practice, and then we have some microvers. So for the first one, you have 6 classes, and you have to try to figure out which class catalyzes this particular reaction. Frequently there'll be little clues here and there, and this is one that you will get used to seeing as a cofactor that is required. A way to approach this is to look to see what has changed from here to here. So what has changed? I've got a double bond that is formed. When that double bond is formed, something has to leave. What has left?
Hydrogen.
Hydrogen. Okay. So I have to lose basically a hydrogen from there and a hydrogen from here in order to make that double bond. So what class of enzyme will this be? Oxidoreductase. So don't call it a lyase, because it's not. At first it might look like an elimination, but you're eliminating hydrogen, and that it always is going to fall under oxidoreductase as a redox reaction. NAD, when you see it like this, where it's being reduced, so my NAD gets reduced, my substrate here is getting oxidized. Whenever you see an NAD cofactor, it's an automatic signal that that's an oxidoreductase that you're looking at. Okay, because those electrons and hydrogens have to go somewhere. So that's an oxidoreductase. Okay, so for this one, again, you have to kind of look to see what changes from my reactants to my products.
Transferase.
Okay, so that amino group became a carbonyl, and then I have a carbonyl here that became an amino. Transferase, transferring functional groups between two molecules. Okay. Then I have to look to see what's changed. You also have to be careful because sometimes not all the reagents are shown.
Hydrolase?
It is a hydrolase. Yes, this is a hydrolase. Okay, I didn't put water over the arrow, but when you look at what you started with and what you end with, you gain an OH and an H. Okay, so I've gained water. And not only do you need to be careful because they might not necessarily show any of the inorganic things coming in or leaving. You also have to be careful that if you see water over an arrow, you can't automatically assume it's a hydrolase because water is also involved in eliminations and additions of where lyases are working, so you just have to be careful. But yeah, what kind of functional group is this right here? Ester. And if I hydrolyze an ester, I make
Carboxylic
acid
and alcohol.
and alcohol. Okay. This is one that we do second semester. We'll talk about this one.
Oh, of course.
So that is a 1,6-fructose bisphosphate.
So just oxidoreductase. I think it's an oxidoreductase. It's removing hydrogens. acid removes the, well, okay, it turns the 4-carbon alcohol into a carbon ion, takes the hydrogen, puts it on.
It is a lyase.
Oh,
Yes,
it is a lyase.
Sometimes it helps to actually look at this in a linear form. Not to help, because you're basically taking fructose and cutting it right down the middle. When you end up breaking the carbon-carbon, so well, it'll be one carbon oxygen and one carbon-carbon bond that gets broken to break it into its two pieces, then those electrons do have to go somewhere, so you end up forming those kind of carbonyl-looking structures. But this one is tricky because we are basically cutting a bond there and we're cutting a bond, let's see, there, in order to get my two pieces. So it is a type of elimination where I eliminated this whole thing over here. But like I said, lyases are usually the hardest to see, and the reactions that you look at for them are the most diverse. So if you can't easily stick it under anything else, it's probably going to be a lyase. But we break the carbon-oxygen, we also break the carbon-carbon single bond. Anything that's breaking carbon-carbon single bonds, lyase. How about that one?
Transference.
You have to look and see what changed. I have a functional group that changed, so I have this, and I go to that.
So transference.
That is an oxidative reductase. It went from alcohol to an aldehyde. Oxidative reductase. All right, eye clickers. So what is that?
I don't know, I can see it.
Oh, it's a little split. Majority wins though. Okay, that is an isomerase.
It's like nasty, I saw this.
Glucose 6-phosphate to fructose 6-phosphate. We already talked about how you can get that keto-enol isomerization between glucose and a fructose structure. So these are isomers of each other. And if you actually took the time to count up all the atoms of each type, they will have the same molecular formula. Okay, so what type of isomer are they? Constitutional. Okay, next one. Oxidative reductase has it. Okay, so what functional group? Carbonyl going to alcohol. I am oxidizing or reducing? Reducing it. Okay. Not the names or the names.
Yeah!
Oh, that's right. Yeah.
We're all
So keto, two different molecules. We just swapped them and
just that.
group is this? What class compound?
Ester.
Ester. And I'm adding water to it and I'm breaking into a carboxylic acid and the alcohol. So that is a hydrolase.
Oh, sad face.
I know, the one person probably didn't see that ATP was getting hydrolyzed there, right? So that's a ligase. Consume ATP to join two things together. In this case, I'm joining L-tyrosine for transfer. Okay, last one. Okay. Oh. Alright, majority wins.
Yay.
Here's the case. I'm adding water. Don't assume that's a hydrolase, because you have to look to see what's happening when I add that water. Okay. I have a carbon-carbon double bond. One carbon got the OH, and the other carbon got the H. That is an addition of water across a carbon-carbon double bond to break a carbon-carbon bond. That is a lyase. Okay. Lyases are very tricky. Okay.
I have a question. You said usually the hydrolase will break into two pieces.
Usually.
Will we be seeing many at all that
Don't. When it doesn't, it's usually because it's a cyclic compound, like a cyclic
Oh,
ester,
okay.
and you break it apart.
Okay.
So it's still one molecule, but it's still one cyclic.
True.
Okay, I, U, B, and B. This is the International Union of Biochemistry.
Molecular
Thank you, Sam.
biology.
Molecular bio. Okay, this is, IUPAC is chemistry and physics. This is biochemistry and molecular biology. It's the same thing. There's a committee somewhere in some room that comes up with how to name and classify things so that we're all using the same language. So these assign systematic names to enzymes. I'm not going to ask you to write systematic names. You have to be able to glean some information from a systematic name if you're given one. There are common names that you will need to know, but we will, introduce those as we go along. If you do propose a name, and I think on the sapling there are some questions coming up that they ask you to propose the name for an enzyme. A good name should tell you the class of enzyme, the identity of the substrate, and the bond or functional group being acted upon. Okay, so a good name should tell you those three things. Systematic names tend to be long. And because they're long, they're awkward to use, and so nobody uses them unless they're trying to very explicitly say what enzyme they're talking about. More often than not, people will use the common names because they're shorter and easier. With an enzyme commission number. And those two things together will explicitly identify your enzyme. So we need to talk about some enzyme class condition numbers. So an example of one of those would be an EC 3.4.17.1. And that happens to be the enzyme class number for carboxypeptidase A. Okay, EC tells you is the enzyme commission number. The 3 tells you the enzyme class. So what was number 3 on our list? Hydrolase. So this is our enzyme class. That's the only thing out of the enzyme commission number that you need to know. Because the rest of it, there's no way you're going to memorize the rest of it. Absolutely no way. But the first 6, if you know the first number and the enzyme class number, you know what class of enzyme it is and what kind of reaction you're catalyzing. So 3 is hydrolase. I'm going to write down the others just so that you know what they were. So 4, in this case, tells me that you're acting upon peptide bonds. The 17 tells you that it falls into a class of metallo C-terminal peptidases. So it requires a metal cofactor. And then 1, that last number is a unique identifier for that specific enzyme. Okay. So, class of enzyme, what kind of reaction, what kind of bond or functional group it's acting upon, and then I get into this kind of subclass that's really quite specific. There might not be very many things underneath this category, this metallo peptidases, that kind of C-terminus. And then that unique identifier is just for it. And that way, if someone's talking about a carboxypeptidase A, or if they use a common name for it, as long as they give that enzyme class number, everyone knows exactly what enzyme they're talking about. Okay. These things show up in lots of different bioinformatic databases. I like the XKZ database because I tend to use that one a lot. So if you're interested in going and looking at these, www.xkz.org is my favorite bioinformatics site. It has lots of bioinformatic tools, one of which happens to be an EC database. I can go look all these up. So maybe if you're a W student, and maybe you're working on a paper right now that might be about a particular enzyme, and if you want some more information, some more references, you might go look up your EC number. Go do it. Okay. I want to just mention kind of an example of a vague name.
That's exciting.
Catalase. A common name for an enzyme. Can you tell what it does by its name? It catalyzes something, right? I don't know anything. It was called catalase because it was such an efficient, great catalyst for doing something, but unless you know what that something is, maybe you don't know. So, it turns out that its EC number, so you're going to be able to tell me something from this, is a 1.11.1.6. And if nothing else, you can tell me the kind of reaction it's catalyzing. Oxidoreductase. Okay, so it's a redox reaction. Do you happen to know what the redox reaction is? Not from that, but if you went and looked up the 11, subclass 11, subclass 1, and then the unique identifier 6, if you went to that database, you'd find out. A catalase actually catalyzes the decomposition of hydrogen peroxide. And that is an oxidoreductase reaction because basically you're eliminating oxygen from your hydrogen peroxide. But common names, yeah, they might tell you something, they might not, but you can look up anything from the EC number. enzymes bind to substrates via some nice non-covalent sort of interactions. So what do you think falls in there? All my,
Hydrogen.
yeah, okay. Intermolecular forces, hydrogen bonds. Okay. Van der Waals, so rather than calling them Van der Waals, what we're really calling these things are hydrophobic. Hydrophobic interactions. They actually mean different things, but it's frequently the same sorts of groups that experience those sorts of forces. So hydrophobics are important. Electrostatics are going to be important. Something that we talk about with protein structures, and so you can have charge-charge interactions. I could put dipole-dipoles on there, but they're not very strong, so I'm not going to. Yeah. The binding site, this is going to be complementary to and interacts with the substrate. And I need to make a point here that the binding site may or may not be the active site. So you don't want to use those two terms interchangeably. The active site is where the chemistry happens. So sometimes they overlap, sometimes it's exactly the same thing, and sometimes they are totally separate from each other. But the binding site is whatever interactions you need to recognize and bind to your substrate through those different intermolecular forces. This is often a cleft or a pocket on the surface of the enzyme. And it's going to have those complementary interactions. And in our cartoon, all those interactions are just perfect. So here's my enzyme, there's a pocket there, and I have some nice little puzzle piece kind of interactions going on. So H represents hydrophobic, so hydrophobic to hydrophobic for my substrate to my enzyme. There's an NH that could hydrogen bond to an oxygen in my enzyme. There's a negative charge in my substrate, where I can get a nice charge-charge interaction with a positive charge in my enzyme. There's some more hydrophobics, and then here's another hydrogen bonding interaction that they set up. So you get this kind of complementary set of interactions in your binding site. Of course, the world never looks quite that perfect. There are two models for binding. A lock and key model and an induced fit model, and like all models that you ever get, one may represent a particular system better than another model, but it depends on the system. So, more often than not, we see an induced fit model, but there are examples out there where it looks like a lock and key. Okay. So, for a lock and key, you have a very rigid binding site for the enzyme, and your substrate is just exactly the right shape to fit into it. And so your enzyme's the lock, your substrate's the key, and it's an exact fit. In an induced fit model, you've got this binding site that actually adapts a little bit to fit your substrate. So we see a little bit of structural change when it binds. Frequently with induced fit model systems, you set something up where you might be able to recognize more than one substrate and act on more than one substrate. And so, like I said, the specificity for your substrate, it could be very, very strict or stringent, or there might be some flexibility there on what it can bind and recognize to. So my binding site, we want to say that recognition of substrate may be very strict, or the word that people like to use here is stringent, or may have some flexibility, and it just depends on what system you're looking at. But unlike chemical catalysts, usually chemical catalysts, other than a particular functional group, it doesn't matter. Whereas with enzymes, it can be very specific. We'll only work on one molecule or one enantiomer of a particular molecule. And then we have two models for binding, it's done on paper. One is the lock and key. Then here we have rigid substrate and active site, or binding, whichever one it is, or both. And then the second one is an induced fit. Fit model, and that's where you might have a binding site or an active site can adjust to fit the substrate. So, kind of back to this idea of stringency. Some enzymes, again, might only work on D-alanine and nothing else. Works on D-alanine, so as soon as you throw an L-alanine on it, it can't work like that. Some enzymes, it might just be that the functional group is specific, but the rest of the molecule doesn't really matter. So for example, we have an alcohol dehydrogenase in our liver. Alcohol dehydrogenase's job is to dehydrogenate alcohols. If you imbibe, then that is the alcohol that you are dehydrogenating, that you have to get it into a form that can be metabolized. When it gets dehydrogenated, you turn it into an acetaldehyde, which is okay. We can actually funnel that into some different metabolic pathways. But if you have methanol, it can't distinguish between ethanol and methanol. Methanol it acts upon, and then you make formaldehyde instead, which is why methanol is poisonous. That's why it's very toxic. It's also an enzyme that can be, since it acts on two different substrates, you can actually outcompete it. And if someone were to go to the emergency room with methanol poisoning, the treatment is to get them drunk. Because if you outcompete the methanol with the ethanol, then it gives the chance to actually let the kidneys excrete the methanol, and then you don't turn it into formaldehyde, and you don't go blind or die from it.
That's what happens if an animal drinks antifreeze too.
Yeah. But it's
Because it
not
tastes really good.
Not methanol, it wasn't antifreeze, it's a glycol instead, ethylene glycol. Okay, so we'll stop there.
Antifreeze